Good afternoon. Yeah. Let's go back to the lecture then on health effects. We had talked about particulates and gave some definitions of particulate matter. What I want to look at now is how does the body respond to particulate matter? Basically, the process by which particulates are removed from the lungs is referred to as clearance, and there's really two different types of clearance. One is mucociliary clearance, and you saw the scanning electron micrograph showing the cilia on the cells of the bronchi. And because of that sweeping motion, and along with the mucus that exists and coats the cells in the lower respiratory tract, that cilia essentially sweeps the particulates out of the respiratory system and carries them to the larynx. Now there they can be either you can cough them up, so you're removing it by physical means. They can also move into the digestive system. Then we have alveoli clearance, and here very, very small particles can be removed during the gas exchange in the alveoli. Alternately, they can be absorbed through respiratory epithelial. So it's actually an absorption process. They can also be ingested by macrophage, and we'll look at that next in just a bit. So what I want to just talk a little bit is where these particles deposit, and I'm going to switch to the next slide because this will give you a better indication of how these particles are removed. Let's just switch to the highlighter. So if we look here at the nasopharyngeal region, just color code that line. So we're looking at this yellow line. What you note is that the very large particles are predominantly removed in the nasopharyngeal region. These are removed by inertial impact, and that material tends then to move into the digestive system where it can be detoxified and removed. Very small material here is removed, is soluble, and tends to move by dissolution into the bloodstream. So here we're looking predominantly at the digestive system. Here at the very small particles, we're looking at removal in the bloodstream. We can also look at the pulmonary system. Here, we'll use the blue line for that. And here you see that there is some removal of these larger particles, predominantly removed by the cilia in the bronchi, and the much smaller particles here tend to move into the alveoli, and that's where they're removed there. And then lastly, we have the removal in the tracheobronchial. So we're looking really at the upper respiratory system here. We use an orange for that. And you can see that here we talked, for instance, of smaller particles being removed, being solubilized and moved into the nasal turbinates where they can then be removed. Some of that those larger particles mention the deposition in the larynx, and that's where these larger particles would be removed. It gives you some sense of the differences in terms of how these particles are removed. That also affects the target organs, whether it actually gets into the bloodstream and then can move into the lymphatic system, whether it moves in the digestive system, whether it hits the liver, okay, or for instance, stays in the larynx. Now, I mentioned phagocytosis. So this is a short video. I'll just play this first.
In phagocytosis, phagocytes are attracted to the area of invasion by chemicals of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So I wanted to play that just so that you have some sense of how these particles are actually removed from the lungs. So for instance, if you're in a highly polluted environment, you're in a work environment, for example, where you have a lot of silica dust that can that you breathe in and can actually reach the alveoli. These phagocytes are important in terms of detoxifying the respiratory system, removing those particles so that they can then be removed and then detoxified. So for instance, these phagocytes can transfer these pollutants to the lymphatic system or to the blood system. Then it can be transported to the liver, where it can be further detoxified. The last thing that we don't want is we don't want these particles to be permanently retained in the alveoli. What you see on the right is an image from a lung of a person that had black lung disease. What you actually see, these black deposits are actually alveoli sacs that have been destroyed. And what's left behind is this black soot. About a third of deaths caused by lung disease, such as emphysema, are the result of this type of deposition of material. Now, the effects on the respiratory system can be both acute or chronic. So for instance, with acute effects, so these are short-term, typically at high concentrations, that can lead to constriction of the airways, so they tighten, can't get as good airflow, which means you don't get gas transfer. Can also lead to edema, which is swelling or release of water from the cells out into the lungs, and can result in secondary infection. Longer term effects, so more chronic effects, can result in cell damage. So this results in necrosis, which is the death of a cell, can result in increased permeability. So that can result in inefficient air gas transfer and also in edema. And then we can have both chronic or acute effects where the pollutant itself, for instance, pollen, causes the excitation of nerve endings, causes the bronchiole to constrict, causes, if you know anyone, for instance, with asthma, causes that wheezing sound. It's because the air is constricted and it's not able to flow into and out of the respiratory system. Pulmonary fibrosis and oncogenesis are both results of chronic exposure. Pulmonary fibrosis, two examples are silicosis, and the other is asbestosis. So silicosis is caused by chronic exposure to silica dust. And asbestosis is chronic exposure to asbestos. Those of you that have been are civil engineering students and have taken CE 312 and 337, even before we went remote, there were some changes in the way some of those labs were conducted, and that was the result of more stringent regulations on silica dust. And because of those regulations on silica dust, changes needed to be made in order to ensure that the exposure to that silica dust in the labs was sufficiently low in order not to be of risk. Oncogenesis is the result of changes in DNA resulting in cancer. So, for example, xylene, trichloroethylene, both are insoluble chemicals. The target organ is the liver, and these chronic exposure to these chemicals can result in liver cancer. So the effects on the respiratory system can be relatively localized, or they can be much more systemic in the body. They really target organs, for instance, such as the lymphatic system or the liver, and can result in cancer. Any questions about the health effects on the respiratory system? If not, what I'd like to do is end this part of the lecture. We're going to switch. So let me save this. And I am going to switch now to